So thank you very much for inviting me to this great meeting. I'm really looking forward to share our data on the analysis of T-cell immunity and development of a peptide-based vaccine to combat COVID-19 with you. So my lab is in non-COVID times focusing on the development of peptide-based cancer immunotherapies, especially cancer vaccines. And these vaccines are based on HLA-presented tumor-associated peptides, as they are presented on tumor cells and differ from normal cells. And usually these tumor-associated peptides are then recognized by tumor-specific T cells, which leads then to the destruction of the tumor cells. And usually we use these HLA-presented peptides to develop cancer vaccines. So we have the possibility to synthesize these peptides in the lab and use them for vaccine composition. The vaccine is then applied subcutaneously into the skin of the patients, where the peptides are uptaken by antigen-presenting cells, followed by priming of T cells by these antigen-presenting cells in the lymph node, and subsequently proliferation of peptide-specific T cells that then could recognize the respective tumor peptide on the cell surface of a tumor cell and destroy the tumor. So this is what we have done before the COVID pandemics. And with the arising of the new virus, we decided to focus on the development of such a peptide vaccine for the induction of SARS-CoV-2 specific T cells for COVID-19. So this is based on the knowledge, and I think you all are aware that T cells play a central role not only in the defense of cancer, but also in the virus defense. Here we have CD8 T cells and CD4 T cells that play an important role, while CD8 T cells could directly lyse virus-infected cells and act as memory CD8 T cells. CD4 T cells have even more responsibilities, so they also act as memory, as a memory pool. They could activate and support CD8 T cell response by cytokines and cell-cell contact, and very importantly, they could improve the antibody production by B cells, also by cytokines and cell contact. And it was also shown that CD4 T cells are able to directly lyse virus-infected cells. So based on this knowledge, we started, I think, in the early February 2020 to think about T-cell immunity in SARS-CoV-2. At this time, nothing was known at all, but we had some information about previous zoonotic coronaviruses, SARS-CoV-1 and MERS-CoV, that could be found in the literature, that showed us that T cells seem to play an important role in coronavirus infection, as it was shown that CD8 and CD4 T cell responses actively contribute to viral clearance and prevent severe pulmonary pathology in SARS-CoV-1, while antibody responses can enhance or even trigger severe illness. Furthermore, there was early upcoming data that virus-specific memory T cells persist for several years after infections. There was one paper published in Nature that showed SARS-CoV-1 specific T cells even 17 years after infection in convalescent donors. And based on these data, we decided to analyze the role of T cells in SARS-CoV-2. And to start this project, the first important question was which structures of SARS-CoV-2 can be recognized by T cells? So what are the T cell epitopes of SARS-CoV-2? To answer this question, we performed computer or algorithm-based prediction of potential T cell epitopes covering all viral proteins and the most common HLA allotypes in the European population. Based on this prediction, we then got 100 CD8 potential CD8 T-cell epitopes for the 10 most common HLA allotypes, and we further added 20 broadly applicable HLA-DR peptides that were predicted to bind to multiple HLA-DR allotypes and were thus broadly applicable. To now prove that these predicted peptides were real SARS-CoV-2 T-cell epitopes, we analyzed a large cohort of COVID-19 convalescents and screened their blood for T-cell responses against these peptides. Based on this analysis, we were able to identify several dominant and subdominant SARS-CoV-2 CD4 and CD8 T-cell epitopes. And we were able to show that T-cell responses against these T-cell epitopes were also present in convalescents that had no antibody response. So convalescents that were seronegative. We further could show that the diversity of T-cell response, which means the broadness of a T-cell response and the number of different epitopes that are recognized by one donor was associated with the mild course of COVID-19 disease, while the intensity of T-cell responses could not be correlated with disease outcome. We further, and in the beginning, screened just for a negative control. Also donors whose blood was frozen before the beginning of the pandemic, with the aim, as I said, to have a negative control, because we did not expect any immune responses in these donors. Unexpectedly, we could also detect T-cell responses against our SARS-CoV-2 T-cell epitopes in these unexposed donors. We were able to detect these T-cell responses against our so-called SARS-CoV-2 cross-reactive T-cell epitopes in 81% of all unexposed donors. And we could show that these T-cell responses are based on similarities of our SARS-CoV-2 T-cell epitopes to common cold coronaviruses, based on so-called heterologous immunity and cross-reactive T-cell recognition. So it's really important to note that the role of these cross-reactive T-cell responses is still not finally clear. So it's not known if and to what degree these cross-reactive T-cell responses protect from infection or have an influence on the course of COVID-19 disease. There are a lot of prospective trials ongoing to evaluate the effect of these cross-reactive T-cells on immunity and disease outcome. We further, and based on my regular research work in the field of hematology and oncology, we're also interested how are the SARS-CoV-2 immune responses in cancer patients, as patients with solid tumors or hematologic malignancies, as these patients were reported to be at special risk for a severe course of COVID-19. Therefore, we analyzed a large cohort of cancer patients that had no contact to SARS-CoV-2 for pre-existing cross-reactive T-cell responses. And what we could show is that the prevalence of pre-existing T-cell responses was markedly reduced in patients with hematological malignancies compared to patients with solid tumors or healthy volunteers. We further could show that this reduced prevalence of cross-reactive T-cell responses might be due to an exhaustion of these T-cells in hematological malignancies. And we further could show that this reduced prevalence is also evident in other viral diseases compared to CMV and EBV. We further analyzed a small cohort of cancer patients that suffered from COVID-19, and we could show that the antibody response in this cohort was comparable to healthy volunteers in the prevalence as well as in the intensity of antibody responses. Furthermore, we could show that the prevalence of SARS-CoV-2 specific T-cell responses was also comparable in cancer patients compared to healthy volunteers, but the intensity of the T cell responses were reduced in cancer patients compared to healthy volunteers. We further could show, when we had a look on single epitopes that were recognized by cancer patients, that the expandability of T cell responses was reduced for several of these epitopes, which means that cancer patients were not able to expand their T cells upon cultivation in vitro. And furthermore, we could show that the diversity, so the number of different epitopes that were detected by the patients was markedly reduced in cancer patients compared to healthy volunteers, again, especially in patients with hematological malignancies. And we could show that these reduced diversity, as we have before shown in healthy convalescent donors, is also in cancer patients associated with a worse disease outcome of COVID-19. We further, and now coming back to the healthy convalescent cohort, we're interested in what is happening with the T cell responses and immune responses to SARS-CoV-2 in the long-term follow-up. And therefore, we monitored some of our initial cohort donors for up to 6 months after infection, and screened them for T cell and antibody response and asked them for post-COVID symptoms. We could detect post-COVID symptoms in about 30% of our donors. Most of these donors included in the trial had a mild COVID-19 disease. Only a small fraction was hospitalized, and there were a lot of people that had almost no symptoms in the initial infection, but later suffered from long-term COVID symptoms. We could show that T cell responses persist after 6 months, with CD4 T cell responses even increased after time, while we could show that anti-spike IgG and IgA antibodies decreased, whereas nucleocapsid antibodies persist over time. Furthermore, we could show, when again focusing on single epitope T cell responses, that there were T cell epitopes that mediate persisting T cell responses, while there were also T cell epitopes which show a decrease in T cell responses in T cells targeting these epitopes. We were able to detect a so-called persistent dominant T cell epitope group that were recognized by T cells after 6 months in up to 100% of analyzed donors. And we further thought that these might be the optimal T cell epitopes and optimal targets for the design of a COVID-19 vaccine to induce T cell responses. So why did we decide to develop a peptide-based vaccine candidate slowly based on the induction of T cells? So this is based on the one hand on the central role of T cell responses in the immune response against SARS-CoV-2 and the course of COVID-19, as I've shown you before, as well as the suggestion that T cells are the central component of long-term immunity. And we aimed to develop this vaccine based on T cell epitopes from various viral proteins, not only limited to the spike protein, to enable a T cell response that resembles that of the natural SARS-CoV-2 infection. And I think this is most important. We want to provide an alternative vaccine candidate for patient group with B cell deficiencies, especially cancer patients. And here, as you might know, lymphoma patients that are treated with perhaps anti-CD20 antibody therapy that lack B cells for several months after treatments and are not able to develop a humoral immune response after vaccination or natural infection. So how could we manage to develop this peptide-based vaccine in the academia? So we have a preclinical development department for immunology where we do all these preclinical analysis, which I showed you before, where we could do the vaccine design, and we have the option to produce peptide vaccine products in GMP quality to evaluate these vaccines in clinical trials. And we have the CCU Translation and Immunology, which is the department that is performing the translation, that is development, the study concept, study implementation, and finally the conduct of the study. And we have this platform developed in the beginning for cancer vaccines, and could now in COVID times use this platform that was already established to develop a vaccine candidate in a timely manner. So how did we design the COVAC-1 peptide vaccine candidate? COVAC-1 consists of 6 HLA-DR T-cell epitopes that cover multiple HLA-DR allotypes and could therefore be used allotype independent. Furthermore, COVAC-1 and the HLA-DR peptides include embedded HLA class 1 SARS-CoV-2 T-cell epitopes to enable also an activation of CD8 T-cells. And furthermore, we selected, as I told you before, those T-cell epitopes that mediate persistent long-term T-cell response after natural SARS-CoV-2 infection. So as all vaccine candidates need adjuvants to induce strong and long-lasting immune responses, we use also an own developed adjuvant, a TLR1/2 agonist, which is called XS15, which was developed by the head of the Department of Immunology in Tübingen, Hans-Georg Rammensee. The vaccine and the peptide and the adjuvant are emulsified in an oil emulsion with montanide. And we had data from cancer patients as well as from first healthy volunteers that showed that this combination is able to induce strong and long-lasting T-cell responses. So COVAC-1 vaccine design and production, as I told you before, was done at the University Hospital Tübingen in our own GMP facility that has the approval for the production of peptide vaccine products for clinical studies. And in November last year, we could start our first phase 1 clinical trial to evaluate the safety, tolerability, and immunogenicity of COVAC-1. So COVAC-1 was applied once to the healthy volunteers that were included in the study, followed by a safety and efficacy follow-up. So this was the study scheme. In the first part, we included young and completely healthy adults aged 80 to 55. And in the second part, we were able to include patients that were older than 55 and were allowed to have a few pre-existing diseases. So I would like to show you first preliminary data from the younger cohort until day 28, which is the end of immunogenicity follow-up. So as expected for peptide vaccines, including montanide, all patients developed a granuloma at the injection site where the immune response is going on, as well as erythema and swelling. Due to the fact that this immune response is going on locally, we could not detect any systemic inflammatory side effects like fever or shivering or anything like this. And we also did not observe any allergic reactions, so that in total, in this preliminary data, the vaccine is very well tolerated. Concerning immunogenicity, we could induce T-cell responses in 100% of vaccinated subjects after one COVAC-1 vaccination. We could show very early vaccine responses in 92% of donors on day 14 after 2 weeks of vaccine. And we could show that the T cells that were induced were multifunctional, and we could also induce, beside the CD4 T cell responses, CD8 T cell responses. Furthermore, when comparing the COVAX-1 induced T cell responses to the T cell responses observed in convalescent donors, we could induce a much more intense T cell response. And when comparing the COVAX-1 induced T cell responses to published data of the approved vaccine candidates, we could also see that we induce a much higher frequency of SARS-CoV-2 specific T cells, here shown in red, compared to the approved mRNA vaccines or the vector vaccine from AstraZeneca. So what is going on? We are currently evaluating the final results of the phase 1 clinical trial and hope to publish the first data in the end of July this year. And we are now heading on. And just last week, our follow-up study that will evaluate COVAX-1 in patients with congenital or acquired B-cell deficiency, especially in leukemia and lymphoma patients with B-cell deficiency, will start in July this year. So the study setup will be comparable to the phase 1 study. We'll start with a phase 1 part to evaluate if the immune responses are as strong as seen in healthy volunteers. And if that is the case, we will proceed to the phase 2 phase. Otherwise, we will add a second vaccine of COVAX-1 after 43 days to improve T cell responses. So as I said, the study is approved by the regulatory authorities, and we hope to start recruitment in July. With this, I am at the end of my talk. I would like to thank you for your attention, and I would like to thank all the people involved in this project, from the Department of Immunology as well as from the Translational Immunology, and especially my group, which worked in this last year, I think more than double, to still work on the cancer projects and add the work on COVID-19. Thank you, and I'm happy to take questions.